students, I hope you all are fine. So welcome back to the study portal. So today we are going to discuss about immunochemistry. What is immunochemistry, right? So the objective of this class is to understand the structure and the function of the antibodies, how they are used in diagnostic and therapeutic applications. So what are the antibodies, what types of antibodies we have, and how we can use in diagnostic and therapeutic applications, right? So next point is to understand what are the basic principles of the immunochemistry, right, including antigen antibody reactions. So what are the antigen antibody reaction? When an antigen binds with antibody, then what types of reaction? This means your agglutination reaction, proton phenomena, right? Proton phenomena, right? And your CFT, your CRP, right? So these are the agglutination, right? So these precipitation, flocculation. So these are the different types of antigen antibody reactions that we have to be used in this, right? So what is immunology? So immunology is the study of the immune system and the immune system of vertebrates. Vertebrates means ourselves, right? So immunities means exempt, free from burden, right? Every disease is free from we can free from our diseases, right? So that will depend upon our immunity, that how the immunity will works, right? So immunity involves the resistance show and protection of offered by host organism against the infectious diseases, right? What are the three silent feature of immunological reaction? That is the recognition of self, specificity, and memory of response. So these are the main three immunological reactions features. These are recognition of self, specificity, and memory of response, right? Next one is your what is antigen, right? So antigen is any substance which invokes an immunological response, right? So any of the thing that we have immunogen, right? So anything that evokes our immunological response is an antigen. So that certain components of cell membrane act as specific antigen. So they have a specific antigen can bind with specific antibodies. So most of the antigens are proteins or large polysaccharides from foreign organism, right? So most of the antigen are from your polysaccharides or your protein, right? Next is what is epitope. So epitope is a small part of an antigen that interact with an antibodies. If antigen is triangle in shape, then it will have a small circle like epitope that bind with antibodies, right? So this will be your antigenic determinant sites, and any given antigen may have several epitopes, right? Each epitope is recognized by specific antibody. As every has have a single antigen have bind with single antibody, and in same way each epitope is recognized by a specific antibodies, right? Next is your epitope antigen region that interacts with antibodies. So this will be your antigen, right? So this will be your antibody A, antibody C, and antibody B, right? So this will be your antigen binding site where antigen can bind. This will be your antigen binding site where antigen can bind. So this will be your epitopes antigenic determinants. Next is your antibodies. So what are antibodies? So protein that recognize and bind to a particular antigen with higher specificity. When a protein will bind with your particular, every antibody have particular antigen that bind with higher specific, have higher energy, right? Produce in response to exposure of the antigen, right? So one of the viruses, microbes have several antigenic determinant sites to which different antibodies may be bind. So right, so each antibodies has at least two identical sites that bind with antigen, that will also known as antigen binding sites, as I told you in this picture. So belonging to the group of the serum protein are also known as immunoglobulins. So from this antibodies we can also form, from where this will, they will from a plasma cells, right? Serum cells, right? So from belong to which group proteins? So this will called your immunoglobulins, right? So as you can see in this picture, this picture one is the structure of antibodies, and this will be your antigen binding site, right? So in this picture, both this will be your antigen binding site from where antigen can bind. This will be your light chain, so they can bind together by disulfide bond, right? So they have variable chains, right? So they have constant which we have support in this. So these have this region of high and light chain, and this will be a variable region of the light chain, right? As you can see in this picture B, so this will be your antigen, and they can bind with antigenic determinant, and from where which type of site they will have, they have antigenic binding sites. From where they will bind from heavy chain and the light chain, right? What is immunoglobulins? So what are immunoglobulins? So immunoglobulins are the molecules, right, so that can bind with serum proteins, or that can bind with antigen, right? So this will be a heavy chain. They will bind from your disulfide bonds. This is variable chain, this is constant chain, right? So what types of immunoglobulins we have? We have 5 types of immunoglobulins. That will be your IgAg, IgM, IgGd, IgA and IgE. IgA means alpha, IgE means epilepsin, IgM means mu, IgG means gamma, IgD means your delta. Right? What are immunoglobulins? So, a specialized group of proteins have mostly associated with gamma globulins fractions. So, from where they will bind? So, they will bind through. So, they will bind a specialized group of proteins. They are mostly associated with gamma globulin fractions. And immunoglobulin is a functional term, while gamma globin is a physical term. Immunoglobulin is which type of term? This will be a functional term, and gamma globulin is your physical term. What are the abbreviation we can use for this? Which this will we can use for is IgG. IgG are produced by plasma cells. As I told you, immunoglobulins, right? Or serum cells we can also. So from where they will produce? So they will produce from plasma cell in response to an antigen, which function act as an antibodies, right? So now we can discuss the structure of the immunoglobulins. So all immunoglobulins molecules basically consist of 2 identical heavy chains and 2 identical light chains. So they can held together by a disulfide bond, or held together by disulfide linkage, or interchain or intrachain. We can, or they can bind with non-covalent interaction, right? It is Y-shaped tetramer, means H2L2. Right? So it have 450 heavy chains have 450 amino acids, whereas light chain has 212 amino acids. Right? Heavy chain of IgG are linked to carbohydrates, hence IgG are glycoproteins. Right? Means that heavy chains of IgG linked together by carbohydrates, and IgG are your glycoproteins. Right? What is variable and constant? Right? So each chain of IgG has 2 region. Immunoglobulins has 2 region, right? Upper one is your variable, lower one is your constant, right? So light chain have amino terminal half and carboxy terminal half. From where amino terminal half is also known as variable, that will be your VL, and carboxy terminal half is a constant region, that will be known as CL, right? Heavy chain have 1/4 of amino terminal, that will be variable region H, that is VH, and remaining 3 are constant, that will be also known as CH1, CH2 and CH3. Right? So from where amino acid sequence from variable regions of light chain and heavy chain is responsible for how they will binding with the immunoglobulins with an antigen. So they are the certain hypervariable region interface between the relative invariable regions, right? So light chain has 3 hypervariable region, and heavy chain has 4 hypervariable regions, right? So the hypervariable regions are more specifically determine the antigen antibody bindings, right? So this will be the picture of your structure of immunoglobulins. So this will be your heavy chain, this will be your light chain, right? So they can bind together by NH2 bond, right, ammonium bond, and this will be a binding site. This will be found by hinge region, from where the This will be your carbohydrate region, and upper one is your glycoprotein region, as I told you, right? They can bind through disulfide linkage, right? What is proteolytic cleavage of immunoglobulin? Proteolytic, proteo means protein and lytic means breakdown. How we can done is, so Papen enzyme split the immunoglobulin at the site between CH1 and CH2 in two fragments, right? So fraction antibodies antigen can bind, and specificity will determined by VH and VL, and fraction crystallizable means your complement binding, right? So this will be a Papen binding, as you can, so this will be a CH1 and CH2, this will be your complement binding, right? Another proteolytic enzyme has pepsin cleavage, so they can bind through pepsin, they can divide from the center, so this will be known as FAB2, right? Now come to the immunoglobulin classes. So what are the immunoglobulin classes? So means what types of immunoglobulins we have, that is IgG, IgM, IgA, IgD, IgE. Ig means immunoglobulins. So this will be a gamma heavy chain, mu heavy chain, alpha heavy chain, delta heavy chain, and epilepsin heavy chain, right? So two types of light chain we have, that is kappa and lambda, right? So we can done is, they can never mix together, right? Kappa is 60% is more common in human, right? Mainly common in human, that is 60% of kappa chains we will have, right? Now come to the immunoglobulin G, that will be gamma globulin, gamma heavy chain, right? So this will consist your major serum bodies, right? So this will consist your 75 to 80% of single Y-shaped unit monomer. It is mainly seen in secondary immunoresponse, right? So they can transfer from blood vessels easily, right? So and they can, IgG can cross through the placenta and transfer the mother immunity to the fetus, right? Right, so this will be a gamma globulin, right? Rh ISO immunization means this will occur, this will also known as hemolytic disease of newborn. Hemo means blood, and RBCs, and lysis means breakdown. So this will be a hemolytic disease of the newborn, and this will occur when a mother is Rh negative and fetus is Rh positive. So in this, during pasteurization, the fetal RBC can enter into the maternal circulation, and they can lead into the formation of anti-Rh body. So that during the next pregnancy, first pregnancy will be normal, but the second pregnancy will cause your Rh positive. So this will be your, when antibody B being IgG in class can enter into the fetal circulation, causing fatal hemolysis or neonatal jaundice this will cause, and neonatal death or miscarriage they will also cause, right? Anti-Rh means AB injected within 24 hours of the delivery of first child. So then we can done is, for this anti-Rh antibody we can given in through injections within the 24 hours of the delivery of the first child. This will cause a ISO immunization and protect future pregnancy. So this will protect from your immunization as ISO immunization and protect future pregnancy, right? Next is opsonization effect. IgG will cause your AFB can bind with microbes, and also the FC portion or can fix with macrophages. So macrophages is the self-engulfment of the cells, and so that phagocytosis is made easier. So this will be known as your antibody dependent cell mediated cytolysis, right? So in this ADCC, this will be your antibodies serve as bridge between the infective target cells and the effective cells. So this will cause a natural killer cells and secrete lytic enzymes and lysis of infected target cells, right? So as this will be a phagocytosis, and this is also when the antigen will be binded through it, right? This will be an antibody dependent cell mediated cytotoxicity. So when a neutrophil will act on the targeted cells, so the natural killer cell will help and support this. So this will be a surface antigen can bind, and macrophages and eosinophils will be the FC receptors of this target. So from where they will bind with bacterium covered with IgG antibody, this will protect from this. This will also known as ADCC, also known as, right? Now come to the IgM. This is the largest molecule and J-shaped molecule this will be. This is a pentamer, right? So this will be five Y-shaped units that held together by J polypeptide chain. This is pentamer and bind with five antigenic sites. As you can see in the 1, 2, 3, 4, 5. This is J-shaped like due to its large size, IgM cannot transfer into the blood vessel and they restrict it to the blood stream. So they can only restrict it to the blood stream, right? IgM is the first antibody to produce in response to an antigen and it is most effective against invading organs. That may be upcoming chronic infections. This will antibody IgM will help in your chronic infection. जो भी recent infection आपको हुआ है उसमें आपको पता चल जाएगा, right? IgM will cause your invading upcoming infection, they will control, right? IgM is predominantly class of the antibody and produced in the primary response to an antigen. So this will be a natural antibody are in IgM, right? If a person blood group having A antigen will bind with antibody E in his circulation, this will call iso means same. So they can bind with isoagglutinin. So this will produce without any known antigenic stimulation. This is also known as naturally occurring antibodies, right? IgM cannot cross placenta if the fetus, even to have its carrier and incompatible antigen, is protected from the natural antibody of the mother. Now come to the IgA, that is will be your immunoglobulin alpha, have high heavy chain, right? So this will be a single monomer and double unit, means dimer or monomer that held together by J chain. So this will be your in your mucosal surface, right? Means your saliva, your tears, your sweat, your milk and your wall of the intestine, right? Or most predominantly antibody in colostrum. That colostrum is from your mother milk, right? So IgA molecule bind with bacterial antigen present on the body and surface and remove them. So IgA prevents the foreign substance from entering the blood cells, right? So they will help from your surfacial and mucosal surface. This will help, right? So the dimer are stabilized against proteolytic enzymes by a secretory piece. So the secretory piece is produced in liver, right? So they will reaches into the intestinal mucosal cells from where it will combine with IgA dimer to secrete to form the secretory IgA, which is then the they will release. So this will be your secretory piece, then they can bind with J chain, right? How they will form? They will form from epithelial cells of the lumen, enzymatic levels. This will be your vesicles. So from where they will enzymatic level will be secreted your IgA and the plasma will break down and they will form a dimeric IgA. Right? Now come to the IgE. IgE will help in your allergic infection, right? Single Y-shaped monomer and normally present in minimum concentration of 0.3 gram per ml, right? So IgE level are elevated in individuals with allergy as it is associated with the body allergic response. That means your hay fever, asthma, anaphylactic, and ana means self, right? Having allergy to the us, right? So anaphylactic shocks and IgE can tightly bind with Fc receptors on basophils and the mast cell and they will release histamine and they causes allergy, right? This is also known as your hypersensitivity reaction 1, with this will cause at the peak of 30 minutes immediate types of hypersensitivity they will cause, right? Or we can type 1 hypersensitivity, right? How they will allergen will bind with IgE is when allergen is bind with, so they can come back to the granules and they have mast cells and they can be degranulized and they release the granules. So this will cause you allergy and the histamine and the other substance that mediated allergic reactions, right? Now come to the IgD, that is delta. This will show in your Y-shaped monomer, right? Y-shaped unit that may be present in low concentration in the circulation and they mainly present on the surface of B cells. B cells means your bone marrow cells, right? This function is not so known and this immunoglobulin is not so known, so you can right? So what are the major functions of this? IgG will help in your secondary immune response or opsonize bacteria, making them easier to phagocyte, or by complement fixation, right? So to enhance bacterial killing, neutralizing bactericidal toxin and viruses, and they can cross through the placenta. Only IgG can cross through the placenta, right? IgA will find in your secretory cell, that will be also known as your mucosal surface, and prevent attachment of bacteria and viruses to the mucous membrane, and it does not fix any complement, right? IgM will be a primary response, IgG will be a secondary response, and they can fix its complement, and they doesn't cross the placenta. They have cholesterol milk, right? IgD may be found on the surface of B cells, and from where it will have act as a receptor of the antigen, whereas your IgGe means your mediated immune response, with the immediate hypersensitivity reaction they will cause, and they form from the mast cells and the basophils upon exposure to the antigen, and that does not fix any complement, and they have main host defense against helminth infections they will cause. Right? Production of IgG by multiple genes. So IgG are composed from light and heavy chains. So each light chain will produce a three separate genes. That is will be a variable region gene, constant region gene, and joining region. From heavy chain they will separate four gene, that will be a variable region gene, constant region gene, joining gene, and diversity region gene. So these are the multiple genes are responsible for the synthesis of one immunoglobulins, right? Antibody diversity, what is? So a person is capable of generating antibody to the almost an unlimited range of antigen, and humans do not contain millions of genes to separately code for individual immunoglobulins molecules. So the antibody diversity is achieved from two surfaces, that will be a combination of the structural genes and the somatic mutation. Mutation means changes, right? They will cause your paraproteinemia, that will monoclonal immunoglobulin. This will cause you multiple myeloma, Baines Jones proteinuria, heavy chain disease, amyloidosis, hypergammaglobulinemia. Hyper means increased level, low, hyper means low, well-destroyed microglobulinemia. These are the different diseases from where they will cause. Right? Multiple myeloma is also known as plasmacytoma. Right? Plasma cell cancers constitute about 1% of all cancers. Female are most susceptible of these, usually occurring when females have 45 to 60% years. Right? So malignancy of single clone of plasma cell in bone marrow. So this will result the overproduction of the abnormal immunoglobulins, have mostly 75% of immunoglobulin, in some cases of the 25% of IgA or IgM. IgG is a type is not seen in younger adults, so they will be less common, and it is will be more severe, right? So this will be a crab. So this will be your C means calcium is increased, R means renal failure, A means anemia, and B means lytic bone lesion. So these are the different hyperkalemia they will cause. So these are the what types of disease they will cause, right? These are the symptoms of this. So spontaneous pathological fractures of weight bearing bones, ribs, whatever may occur, and proteinemia maybe leads to proteinuria, right? Recurrent infection are more common in normal immunoglobulins is diminished. Raised beta 2 microglobin and bone marrow examination we can done. Malignant plasma cell. X-ray will show your punched out osteolytic lesion. Osteo means bone breakdown lesions they will show, right? This will be cause of myeloma in the center of the bone marrow, where the outer side of the myeloma affected cells, and these are the normal cells, right? So from on Jimson stain, red stain, we can see this type of staining will be shown. Right, this is the active myeloma, how this will cause. So in this will be the marrow, bone marrow, and this will be the lytic lesions they will cause, and this will help in bone. So they will have collapsed vertebrae, and this will be your bone marrow plasma cells. What is heavy chain disease? Heavy chain disease are the some portion of the heavy chain are deleted during the synthesis. That some amount of heavy chain will be deleted during when they will producing, right? So they can join with light chain to normal immunoglobulin. So defective heavy chain is excreted in urine, and gamma chain disease is hepatosplenoma. Hepatosplenoma means enlargement of the spleen, and the lymphadenopathy, and alpha chain will cause your abdominal lymphoma and malabsorption, right? Amyloidosis is really rare. About 20% of patients with myeloma develop amyloidosis. So they will develop deposit into the light chain of the tissues like liver, kidney, intestine of patients. Multiple myeloma. Means multiple myeloma, right? So thank you class for joining this.